Hi, this lecture is from the Miami airport. Today we're going to talk about proteins. If we look at a cell and we dry it out, so we remove all of the water, about 50% of that dry mass is proteins. So what's the rest? The rest are lipids, carbohydrates, and nucleic acids. And we've already talked about nucleic acids and lipids, and I think we even talked about some carbohydrates. So a protein is a polymer. A polymer, remember, is made up of monomers. And it's like a lot of little beads in a chain. And those little beads, those little individual units in a protein are called amino acids. And they're called amino acids because one side is an amine and the other side of the molecule is carboxylic acid, which is that carboxyl group that we've talked about. Proteins are very important. Some of them make up structure. The most important proteins, though, are called catalysts, and they speed up reactions and are very important in regulating the cell's metabolism. There are 20 different types of amino acids that can be scrambled up and mixed in long chains to make different proteins. And so what we're going to talk about now is what an amino acid is and these bonds between the amino acids. This is an amino acid. Notice that there's a carbon here in the center that has 4 bonds. And an amino acid has 4 things that bond to it uniquely. First of all, it has an amine group, if you'll remember this, from the different types of chemical compounds that we have. So it's a nitrogen and 2 hydrogens. It has a hydrogen bonded to the bottom of it. And it has a carboxyl group, the double bonded oxygen with the hydroxyl. And at the top it has what is called a side chain, signified by R. There are 20 amino acids, and in these 20 amino acids, each R is different. In this case, you might have a methyl group for this R, and you have what is called glycine. This is a drawing of 2 amino acids hooked together. You can see one here, it's the central carbon. Here's another one with the central carbon. They both have amine groups. They both have carboxyl groups. Notice there's this special link. Remember, there was a hydroxyl group here, and there was a hydrogen here. Through a mechanism called dehydration, water is removed, just like we studied in the formation of other polymers. And you have what's called a peptide link. That's that bond between the carbon and the nitrogen. We can order amino acids. We want to know what's the front and what's the back. So the end that has the nitrogen on it is called the N-terminus, and the end that has the carbon on the end of it is called the C-terminus. When we spell in English, we have 26 letters of an alphabet. Well, when we want to spell in a protein, we have 20 types of amino acids that can be put in all different combinations to make something totally different. It's what gives variety to these proteins. Another term you'll hear often is polypeptide, and that just indicates that there's a lot of these peptide bonds, a lot of these amino acids hooked together. These polypeptides are spewed out of the ribosome, and it comes out not just as a sequence, but has some structural changes. Some of these proteins will be globular, they'll be round-like. Some of them will be long and fibrous. And how they behave depends upon 4 types related to their structure. So I'm going to go over the 4 levels of protein structure. They're called primary, secondary, tertiary, and quaternary. The primary is just the sequence of amino acids. So the primary structure of a protein is just what amino acids are in a line, in a row, like that, like the beads on a chain. The secondary structure shows some little kinks. It involves kinks and folds in a protein. We call these alpha helices if they're coiled, or beta pleated sheets if they're little folds. And these are due to hydrogen bonds between the oxygen and the nitrogen. Remember how hydrogen bonds form between water? Well, oxygen and nitrogen are both highly electronegative, so anything nearby that has a hydrogen could potentially form a hydrogen bond. The tertiary structure is further folding, and it's the interaction of those side chains. Remember those R groups. So some of those are going to be charged, and a positive one is going to be attracted to a negative one. Now we have the quaternary interaction, which involves the interaction of 2 or more proteins. So you have 2 polypeptides, And sometimes those two polypeptides will fold together and mesh together to make an active sort of protein. And that's the quaternary structure of a protein. Now, activities of proteins can change, and they can change depending upon the pH, the salt concentrations, or temperature. All of these can influence proteins. For example, if you have an egg and you heat it up, that white part of the egg turns this from this clear to this opaque white. And what we're seeing is a denaturation of some of the structure of that protein, and that protein is called albumin. Well, this is Dr. Pomeroy from the Miami airport, and I hope you've enjoyed today's short lecture on proteins and amino acids.